will discuss on the principle of genetics, which is how a specific trait, like a physical trait or some special characteristic, like the susceptible or resistant and the certain type disease, was passing through from one generation to the other. So during the course, we will include the structure and function of genes and DNA, how DNA was structured, what is genes, what is function of DNA and genes, what is chromosome, what is the genome's differences between eukaryotic cell, prokaryotic cell, or a virus. We will spend some time to discuss on genetic variation, on the differences between the genomes of one individual to another. We will spend some time to discuss on the population genetics. It's quite complicated in the preview of the population when we have compared the gene flow in the one population. And we will use some genetic method to analyze a proper protein function, like how genetic mutation can affect to protein structure and function. On gene regulation, how the mechanism subset of gene was turned on and turned off during a specific period of time and during a specific cell line. If we still have time, we will discuss on the inherited disease, how the certain type disease was passing from one generation to another. So on the time schedule, we will have totally 34 hours for our course. We will spend 20 hours for lecture, 4 hours for the bioinformatics practical, and 10 hours for the exercise. Okay, so let's So this information is quite important, so please pay attention to that. So on the final evaluation for each of you, the final result will account by 20% of the attendance, okay, plus 20% of the tutor attendance class, and 60% is come from the final exam. So what is this mean? So for the attendances, we will have 10 sections. So if you attend to the full 10 section of the lecture, you will have 20 points. And if you lack each session, lacking of the attendance check, for each section, you will have a penalty of 2 points. Okay. We will have also a bioinformatics practical training. So, for example, if wait for me. So I guess that all of you have understand how to use Moodle, right? All of you have already had a Moodle account and know how to use the Moodle, your Moodle account. So to the learning material of this course, you can come to my course, the 12 Bio 1.002 Genetics here. You click on this link, it will provide you all the learning material, including the slide for all the 7 chapters and 1 practical chapter. And in here, you will have also the file for the exercise, the course description, and bioinformatics practical training here. So in practical training, we have, so let me check the time schedule. So in here, we have, later on, we will have a practical training from next week, okay? We have practical training here. So in each practical training, you will use this file, the bioinformatics practical training, answer a question on it, and submit on Moodle. So I will explain more on this practical training assignment during the practical course. Okay. And this practical assignment is the necessary condition for you to attend to your final exam. So if you lack of this assignment for any reason, you will not have the right to attend to the final exams. Okay. For the tutor, we will have 20% of the result is the tutor result. For getting this result, you will have 5 sessions of the tutor for 5 chapters. And if you attend to the full 5 section, you will have 15 points. So if you lack of any session on this, you will have a penalty of 3 points for each session that you lack of the attendance check. And besides that, you will call for to handle the exercise, volunteer. So if you handle successfully one exercise on the tutor, you will have 1 point. So of course, you can have to handle it perfectly. That means you not only provide the result, but also provide the full solution. Yeah. Do you have any question? Okay. So if you handle one exercise fully, we provide all the information for the solution understandable, you will have 1 point. So as you calculation, each student will have maximum 5 times to volunteer to handle exercise. So if you attend to 5 sessions and handle 5 exercise, you will have total 20 points. Okay, and the last 60% come from the final exam. So the exam will be provided both on essay question, quiz session, and exercise. And the duration of the final exam is 1 hour. Okay. So first, do you have any question on this assessment? Now question, what is survival kit? Any other question? I will explain about the survival kit later.
Teacher?
Yes,
teacher,
yeah.
Okay. I have a question about the final exam. So what about the essay? What will exactly we will write about?
Well, the essay is an essay question. That means it's not a multi-choice question. It's not a quiz question, okay? So you will have to answer the question I provide you, but it's not a quiz question. It's not a multi-choice question. For example, I will ask, what are you thinking about, I don't know, gen modification food, for example, something like that. Or what is the mechanism of epigenetic? What is most basic mechanism of epigenetic? Okay, it's not. I will not provide us as a quick question. Okay, so the final exam will be a combination of both essay question, multi-choice question, and exercise. Okay.
Okay, thank you, teacher.
Any other question? Okay, so come to the, I think it's the very important question, what is survival kit? So every year, I receive a complaint that I'm an ICT student, I'm a student who study in environmental, I'm a student who study in space. I think that this course genetic is not very useful for me. I have something more important to do in life. Well, I respect you. I respect all of your thinking. I kind of respect the student who understand what is important for him or her and what is not important for him or her. And I understand that you have I don't know, something more important to do. I don't know what is it, but like having a girlfriend, for example. Don't laugh at it, because having girlfriend is quite important during university. Nothing is more noble than the genetic material of you combined together and produce new generation. So you are here to prevent human extinction. If that is not what that's not important, I don't know what is. So anyway, I respect you. I understand that you have a lot of things more important to do in life. So I have prepared this for you. If you want to pass my course at minimum level with minimum effort, and you don't need a high score, a high result to obtain a fellowship or to apply somewhere, you just need enough result to pass my course. So I have prepared this for you. Survival kit. Okay, you can find this file on Moodle in here, survival kit. Okay, you access into Moodle and the file survival kit was in here. For the student who just want to pass my course with minimum effort. So if you don't want to waste time with my course, just focus on the following chapter at the following point. For example, in chapter 1, you only have to study the structure of a nucleotide. What is structure of nucleotide? What is structure of DNA and RNA, and the differences between the structure of DNA and RNA? Okay. The structure of the virus genome, prokaryotic genomes, and eukaryotic genomes, and what is the differences between those genomes? And what is differences between the eukaryotic nuclear genome and the eukaryotic mitochondrial genomes? Okay. In chapter 2, you only have to focus on the mechanism how a DNA will replicate inside the cell nucleus and what kind of enzyme participates in the process. And furthermore, the mitosis and meiosis, and what is the differences in the mechanism of meiosis and mitosis. Okay. And in chapter 3, the transcription and translation process, prokaryotic cell, eukaryotic cell, and the exercise, of course. Okay. Of course, for the student who want to get the high score, the high result, I don't know, 18 or 20, you will have to study everything I teach you, and everything I have not teach you. You have to search for it by yourself from the book or from the internet I provide you. But yes, I mean that if you want a minimum result to pass the course, just focus on those fields. This is the field, the fundamental field that you have to study if you only want to pass my course. Okay, if you are clearly study all this, you will have the minimum result. Okay, enough to pass. It's not a high score, but it's enough to pass. Okay, and if you want a high score to obtain fellowship or to have a good CV to apply somewhere else, you have to study everything. Okay. Any other question? All right. So if you have any question, you can stop me anytime immediately to let me know that you have a problem, any kind of problem or any kind of question. Okay. So if this, I prepare for you a survival kit. So it's very easy for you to pass this course. Okay. If this is still not are you still wondering why you have to study this unuseful subject, genetics? I don't even need it. Okay, so maybe this slide can explain you briefly why this course is put it in the first year, which is the fundamental years. So science right now is different with the science in the 17th or 18th century, right? At the moment, science is combination of different fields to create application, to create a real product. We need a combination of different expertise in different field study. For example, in USDH, we have a small project to create a spatial equipment here to detect the HPV virus. The HPV is causing many fatal diseases related to liver, and to create this equipment, we need to a cooperation between a physicist and a geneticist. A geneticist will create a primer to amplify the genome of the HPV virus, and it will amplify inside here. The physics will create a machine. Yeah, full screen mode. Okay. Yeah, genetic and cellular biology is some homologous field, but genetic is more focusedly on the mechanism, on how DNA and how the information was passing through from DNA, RNA to protein. And cellular biology is more focused on the cell, on the structure of the cell, on different organelles of the cell. Okay, so come back to this equipment. So the physics will make sure that the equipment in here is maintained and it can monitoring the concentration of DNA inside DNA side to detect of the DNA of the virus at the certain threshold. Okay. For the student in ICT, so nowadays, one trendy way of research is focused on the human genome. Okay, so in 2003, as I remember, the first human genome was sequences, and at the time, people, the media say that this is the huge evolution. It is a huge invention of the 20th century when we first decoded the got information, we first decoded the human genomes, and we can decide a human genome now, a human now. But soon people realize that sequencing the human genome is just a first and simple step. The next step, which is much more complicated, much more time-consuming, labor-consuming, and money-consuming, of course, is annotated genome. In detail, to know that which fragment of genome, which fragment of genes is maintain which function. What is function of these sequences? What is the consequences of that sequences? What is the consequences if I have a mutation or I have a variation on this position? So annotation, the genome is very important task if we want to understand the meaning of our genome. And the ICT, the computer science is play a critical role on this. In water department and in water environment field, we have also to understand the genome. What is the difference of the genome of the Salmonella, the E. coli, and other bacteria in the water resource, in energy, in medical science, and food science technology, of course. Let me take a small example. So this is an invention of the paper published on Nature Biotechnology on 2018 on the nanorobotics. So this was applied in cancer target drug. So in cancer, you have the most common therapy is the chemotherapy, when the patient will receive chemo treatment. But the disadvantage of the chemo treatment is when one disadvantage of the chemo treatment is when the chemo reagent inject into the blood vessel of the patient, it will destroy every cell it meets on the pathway. Okay, it will destroy the cancer cell, of course, but it also destroys the healthy cell. So we don't know, we don't want that. To destroy the healthy cell will make the patient suffer. Yeah, of course, I have this slide on the noodles. So the patient will have to suffer from the healthy cell was killed, and this is not what we target. We want to kill the cell that we want to kill. We want to kill only the cancer cell. Okay. So how to prevent that? So scientists have invented a structure called the nanorobotic, in which from a circular DNA of the bacterial strain, the M13, from by a technique called the DNA origami, it will form a plate sheet of DNA fragment like this. And this structure will contain the chemo regions inside of this. Okay, and when it's the different other DNA structure will use as a table to make the plate sheet in here circulate and make a tip shape like this with a chemical region to kill cancer inside. And this structure, called the nanorobotic structure, will be released into the blood vessel, okay, until it will travel in the blood vessel until it reaches the tumor cancer cell. And in this, it will release the chemical regions, and start to open the structure and release the chemical region to kill the cell. So let's watch a small clip on how a structure of nanorobotic was formed from the circular DNA. So as you can see here, the structure of DNA was denoted in the yellow color. We'll use adding to the DNA table is the structure to make it in the plates in a plate shape like this. And the chemical region will be added inside the structure inside here, and it will close the structure to form a tube structure, a tube shape. So as you can see in the same analysis, the structure have the size only around 1 micrometers, and it can carry the chemotherapy into the smallest blood vessel. The more detailed information on this, you can see it in the paper with the title here. So during the course, we will discuss on 7 most related topic of genetics that I have divided into 7 chapters. So for the first chapter, we will discuss with each other what is DNA, what is genes, and what is RNA, and what is the structure of DNA and RNA and the genes. In chapter 2, we will further discuss on the mechanism of DNA replication, how DNA was replicated inside of a nucleus cell. We will discuss more on the transcription, which is the process passing through information from DNA to RNA. Translation is the information transfer from mRNA to protein, and the protein structure, how a protein was formed from a polypeptide. In the next chapter, we will discuss on genetic variation, how the differences, very small proportion differences in the genomes can lead to the variation in the phenotypic outcome. And genetic marker, what is genetic marker and what is the application of genetic marker on forensics science and also in parenting testing. And in next chapter, we will discuss on the classical law of inheritance, Mendel law and Morgan law. You have studied basically on this law on high school, but in this chapter, we will further discuss more deeply on the molecular mechanism of this law. And in the next chapter, we will discuss on a very tricky thing and complicated aspect is the linkage disequilibrium. And we will discuss also on population genetics and the law of Hardy-Weinberg equilibrium in the population that equilibrate. And in the last chapter, if we still have time, we will discuss further on epigenetics, on how a gene and subset of genes was turned on and turned off inside a cell. Okay. So, chapter 1, DNA and RNA. So, what is genetics? So, genetic is the science focus on heredity. Heredity is what is the phenotypic characteristic that a parent can pass it into its offspring. For example, the phenotypic characteristic can be the statute body. Normally, the tall and grandparent will tend to have a tall offspring, or the hair color, or the eye color, the blood group, and sometimes the phenotypic characteristic can relate to the resistances or susceptibility to a certain disease. For example, like type 2 diabetes or heart disease or cancer. So normally, children will inherit their biological appearance from their parent, but based on which mechanism? So first, let's watch a small clip on how appearances differ with each other based on genetic factor.
Unique. Each of us has a combination of characteristics or traits that is different from anyone else's. Some traits are easy to see, like eye color, height, and freckles. Others are less visible, like asthma, musical talent, and disease risk. Some traits are inherited, and some are shaped by the environment. Eye color and nose shape are inherited traits. They are controlled by genes, which we inherit from our parents. The language we speak and the music we like are shaped by the environment, which includes our culture, our experiences, and our physical surroundings. Most traits are influenced by both genes and the environment. For example, tall parents tend to have tall children, but height is also influenced by nutrition. And while diabetes tends to run in families, some types can be controlled by diet and exercise. Even when someone is born with a body type suited for dancing or playing basketball, it takes training and practice to learn these skills. Together, our genes and our experiences make us who we are.
So our appearances is the combination of many phenotypic traits. Study the trait, we know that some trait is mostly controlled by the genetic factor, like blood group. You are born with it. Like eye color, like a certain congenital disease. For example, polydactyly or syndactyly or cystic fibrosis. You are born with it. Some other trait is mostly controlled by the environmental factor, like your knowledge, or like the taste of music or the language you speak. And a large proportion of traits is controlled by a combination of genetic factor and environmental factors, such as the height or the risk of getting type 2 diabetes or risk of getting cancer. Normally, if an offspring have a parent or someone in family having type 2 diabetes, he or she will have a higher risk of getting type 2 diabetes compared to a normal persons. But with because type 2 diabetes is controlled by both environmental factor and genetic factor. So if he or she will have a healthy lifestyle, have a physical activity oftenly, have a healthy diet, he or she can decrease the chance of getting type 2 diabetes compared to a not healthy lifestyle. Okay. So first, let's discuss on some terminology. First is what is gene? So gene is a basic unit of genetic information, of course, but classical theory thinks that genes is a fragment of DNA that encoding for proteins. So in human genome, we have around 23,000 genes like that. I mean, 23,000 genes was coding for protein. But recently, with the development of next generation sequencing, scientists know that many genes are not coding for proteins. They're coding for RNA. They are coding for other factors that control the epigenetic of the genomes. So nowadays, they expand the definition of genes is the fragment of DNA that maintain a specific function. That specific function can be coding for protein, which is a very important component of the human cell. But also, it can maintain the other functions, like control the expression and repression of the certain other genes. So next comes the genome. Genome is the full set of genes inside a cell. So, for example, in human, we have the genome is the combination of 23 pairs of chromosomes. Chromosome is a structure of DNA that store the unit of genes. In eukaryotic cell, chromosome is combination of DNA and a protein called histone. We'll come about it later in this chapter. DNA stands for the deoxynucleic acid. Deoxyribonucleic acid is contain the genetic instruction for every cell in the human body. Some say that DNA is the blueprint of cell, like the blueprint when we want to build a house. The blueprint of the house is telling how high the house is, how many floors the house have, where is the bathroom, where is the living room, where's the dining room, where's the bedroom. In DNA, it contains the information of each protein inside the cell. Different with the blueprint of the house. DNA is like a blueprint of the city. So every kind of cell in our body, like the blood cell, brain cell, the bone cell, the neuron cell, the erythrocyte, white blood cell, and the platelet will have the same DNA background, okay? But they have a different structure because inside here, the blueprint is not only to build this kind of cell, but also contain information to the different kinds of cell. It depends on the cell that this or that subset of gene was expressed at. Okay? We'll discuss further on this aspect on chapter 7. So the transformation of information from gene to RNA to protein was listed in the slide below. So the process of information transfer from DNA to RNA is called transcription. And the process of information of RNA to protein is called translation. And after a translation, we will have a polypeptide composed by different sequence of amino acids. And this polypeptide will be through the post-translation modification to form protein. And protein is play a role in many different processes of cell. It can be structural protein, which can build the cell. It can be enzyme, which catalyze for biochemical reaction in the cell. So the brief consume of molecular of DNA we can see on the clip below.
our bodies to function, we need to supply them with a variety of nutrients we get from our diet. Our bodies cannot use the food as it is when it enters our digestive system. The process of chemical digestion uses different proteins and enzymes to break down the food particles into usable nutrients our cells can absorb. And where are the instructions to manufacture these and all the different types of proteins we need to stay alive? The instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into two processes, transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in three stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. The RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called Intron Splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus for the nuclear product and enter the cytoplasm to begin translation. The information in the mature messenger RNA strand translated into a protein. The nitrogenous bases are grouped into 3 letter codes called codons. 63 codons. These codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Begins with the messenger RNA strand binding to the smaller ribosomal segment upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the metacodon sequence of the transfer RNA. Base pairing occurs between the codon of the messenger RNA and the antecodon of the transfer RNA. The transfer RNA molecule binds to the start codon. The large ribosomal segment binds to form the translation complex, and initiation is complete. There are 3 distinct regions, E, P, and A cells. Individual amino acids are grouped to the messenger RNA strand and transferred to the protein base pair of the codons and lipids. Each molecular transfer corresponds to a particular amino acid. Transfer RNA molecule binds to the A cell and between its amino acid and attached to the transfer RNA at the P cell. From the raw codon to the transfer RNA molecule exits from the A cell, and the A cell is open to accept the next transfer RNA molecule. Contained into the step codon is used.
Sir, are you still here?
Yeah.
I'm sorry, but yeah, as someone have said, the sound is quite distorted, so we couldn't listen a little anything.
Okay.
Top coat of nutrients we get from our
It's
diet.
better now.
Thank you.
Our bodies cannot use the food as it is when it enters our digestive system. The process of chemical digestion
It's
uses
better
different
now, guys. Okay.
Yeah.
Okay.
Does it sound better now?
enzymes to break down the food particles into usable nutrients our cells can absorb. And where are the instructions to manufacture these and all the different types of proteins we need to stay alive? The instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into two processes, transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in three stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called Intron Splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down 1 codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation. The purpose of translation is to produce polypeptides quickly and accurately. After dissociation, the polypeptide may need to be modified before it is ready to function. Modifications take place in different organelles for different proteins. In order for a digestive enzyme to be secreted into the stomach or intestines, the polypeptide is translated into the endoplasmic reticulum, modified as it passes through the Golgi, then secreted using a vesicle through the plasma membrane of the cell into the lumen of the digestive tract. Proteins are needed for most physiological functions of the body to occur properly, such as breaking down food particles in digestion, and the processes of transcription and translation make the production of proteins possible. In order for our bodies to function,
Okay, so the next part, I guess that we will have a small break, 5 minutes, and before we go into the next part, okay? 5-minute break. Thank you. Yes, I will share the link of this video in the end, of course, okay? Okay, so let's come back to our course. Oh, I got a question. The gene which are not between chromosomes. Okay, so actually, this is a very nice question. This is very good question. So normally, if we recognize a genes as a fragment of DNA encoding for proteins, so we know if it's a protein because when we sequence the amino acid, we know that this fragment of gene was encoding for the fragment of polypeptide. So with this type genes, scientists will sequence for the promoter, which is the combination of transcription promoter with the sequence that the combination of transcription factor binding in. So if the gene was not between promoter and terminator, it's called the preserved genes. Tiếng Việt gọi là gì? Giả gen. Giả gen nghĩa là những cái vùng gen mà nó không nằm ở trong cái giữa cái promoter và cái terminator. Thì thực ra là có rất nhiều cái vùng giả gen này. Thì một số giả thuyết cho rằng là cái vùng giả gen này ngày xưa là nó cũng là gen thật, nghĩa là cũng có promoter cũng có terminator. Tuy nhiên trong quá trình tiến hóa thì cái gen này trở nên không cần thiết, thì một số cái đột biến làm cho thay đổi vùng promoter đi, khiến cho cái transcription factor không thể bám được vào đấy. Thế thì trong thực tế thì những cái how much rate of that usually gene là cái rate của cái giả gen này, preserved gene này thì nó phụ thuộc vào từng loài. Có loài nhiều, có loài ít. Ở các loài động vật mà bậc càng cao ấy, thì cái rate của cái giả gen này tương đối nhiều. Và bioinformatics researcher how to remove that data, thì cái giả gen này thì ví dụ ở loài người ấy, thì là cái giả gen thì đã được hầu như là đã được sequence và công bố rồi. Thế còn ở các cái loài động vật mà chưa được sequence nhiều ấy, thì để xác định những cái giả gen này thì đầu tiên là người ta sẽ xác định vùng promoter và vùng terminator đã, đúng không? Terminator thì nó có trình tự rõ rồi, đúng không? Thế còn promoter thì nó tùy thuộc vào cái vùng transcription factor bám vào cái gen đấy là gì, thì người ta sẽ dựa vào những kỹ thuật để xem là những cái vùng nào thì có trình tự như thế nào, thì cái transcription factor nó xu hướng bám vào, gọi là cái motif binding. Các bạn search ở trên mạng thì có cái cụm từ là motif binding ở transcription factor đấy. Thì người ta sau khi xác định cái motif binding rồi thì người ta sẽ xác định được những cái vùng nào có khả năng cao là promoter, và những cái phần gen nào mà không có promoter và không có cái terminator ở cuối thì nhiều khả năng là giả gen. Tuy nhiên vẫn có xác suất, nghĩa là xác suất có thể là sai, bởi vì là khi mà tính p-value, không biết các bạn đã học về cái thống kê, xác suất thống kê thì đã biết rồi, đúng không? P-value nó tính ra thì ví dụ p-value nó bằng 0,001 chẳng hạn, thì mặc dù là rất là có ý nghĩa thống kê nhưng mà vẫn có xác suất là 1% là cái kết quả đấy nó observe là bị sai đấy, cho nên là nó vẫn có xác suất là có khả năng là mình tính sai. Ok. Okay, so come back to a little bit of history. So this is a timeline of genetic, how the genetic was involved from time to time. So, for example, in 1928, Frederick Griffith, scientists, have first performed an experiment on the mouse. He used two strains of bacteria. One is called the rough bacterial type R, stands for rough because the shape of the outside colony is quite rough and have a not smooth shape like this. Okay, this type strain, when he injected into the mouse is still alive. So that means that this type of bacteria is not quite toxic to the mouse. He used another type of bacteria called type S, because the shape of the colony is quite smooth, like this. And when he inject this type of bacteria into the mouse was killed instantly. So that means that this type S, bacteria type S, was quite toxic to the mouse. He carried the third experiment when he using the same type S, but he pre-treated it with heat. So the heat will kill all the type S bacteria. And when he injected into the mouse still alive, because most of the S was killed. And in the final experiment, when he mixed a combination between the type R, which is not toxic to the mouse, and the type S, which is deadly to the mouse, but it had been pre-treated, killed with heat. And the combination to the mouse, he expected the mouse to still be alive because the two type R is non-virulent and the type S is violent, but it treated with heat. But in fact, the mouse was killed. And in the sample of the mouse, he find out that there is in the sample there is a like type S bacteria. So what kind of conclusion we can make from the experiment of Griffith? Can anyone tell me?
I think somehow the type R has, when we combine with the heat kill type S, it somehow turned into live type S, maybe.
Okay. Which kind of factor? So in here, the type S have been killed. But if we inject only the pre-treated heat kill type S into the mouse alive. But when we mix with the type R, some factor, at the moment, Griffith have no idea about the DNA, but at the moment, he knows that there's some factor in the type S has transferred into the type R, and the type R has been using this factor to synthesize the new live type S, and this new live type S would kill the mouse. Okay. So continue with the experiment of Frederick Griffith in 1944. Marliott and McCarthy had performed the same experiment, just a slight modification. So in this experiment, they use the type S, the same strain that is violent with mouse here. When you inject it into the mouse will kill instantly, right? But now they have pre-treated with several substances. So in the first experiment, they treated with the enzyme that destroy polysaccharide. And after we retreated, injected the strain pre-treated with the enzyme that destroy polysaccharide into the mouse would kill. In the next experiment, he inject the type S, which is involved with the enzyme that destroy lipid. The mouse was also killed. In third experiment, he mixed it with the RNase, the enzyme that destroy RNA. The mouse was also killed. In the fourth experiment, he mixed it with the proteinase, which is destroy the protein. The mouse was also killed. And in this fourth experiment, in the sample of the killed mouse, they found the S strain, the live S strain. But in the last experiment, he mixed with the DNase, the enzyme that destroy, cutting through and destroy the DNA. When they inject the sample was pre-treated with DNase, the mouse was still alive, and there's no sample in the sample of the mouse. There's no live H strain was found. So that means what? That means that the DNA was play a key role in transfer the information to build a new live H strain, right? And in another experiment of 1952 that opened the structure of DNA, revealed the structure of DNA, was the experiment of Hercy and Chase in 1952, when they using a virus, a phage that inject into the bacteria. So in virus, we know that the virus was composed by the nucleus is DNA or RNA, in some case. We'll discuss about it later. And the envelope was composed by protein. And in protein, the main one of the main component from the protein was sulfur. So in the first experiment, the virus was coded by the protein that radioactively labeled of the code here in the sulfur. They use the S35 instead of S32. And then they let the phage was injected into bacteria. And the results show that all of the radioactive label was like inside, and none of this protein was injected into the bacteria. In another experiment, this time they was not labeled the protein envelope, but they labeled the nucleus by the phosphor radioactive label. So phosphor is the main component of DNA. And then when they let the phage inject into the bacteria, they see that the label of nucleus component was injected from the phage into the bacteria. So now they find out that the radioactive label was located inside the bacteria. That means the DNA was injected from the phage into the bacteria, not the protein copy, not the protein envelope. So from the three experiments of Griffith, of McCarty, and of Hercy and Chase, in 1953, James Watson and Francis Crick have firstly demonstrated the 3D structure of the DNA molecule. So now we all know about this famous invention of Watson and Crick. So if we ask any biologist, any genetician, even a high school student who was the first invented about the 3D structure of DNA, they also say it's Watson and Crick. But very few people remember about Hercy and Chase. Very few people remember McCarty or Griffith. So that's what science lies. So in science, we can never do it alone. We must base on the result of somebody else. Anyway, Watson and Crick have demonstrated the first 3D structure of DNA, which shape of the helix ladder. Okay, with the two sides of the ladder was the sugar and phosphate backbone, and the each level of the ladder was a combination of base matrix of A, T, G, and C for the checkup rule. So since 19 1953, that the DNA model was discovered by Watson and Crick. In 1977, the first sequencing machine was invented, followed the Sander sequencing machine. It based on the DDMTP, which is when it's combined to the synthesized DNA sequence, it will stop the sequence right there and provide a signal. So from 1977, when the first sequencing machine was invented, with the same year, the first genome was sequenced, about 5 kilobases each genome from a bacteria. In 2003, the first human genome was sequenced, about 3 billion base pair. At the time, I don't know if 2003 you guys was born, but at the time, people and media say that this is the huge step of human. When we reveal the code of life, when we reveal the code of God, now we can decide human, we can design human as we wish. But soon they realized that sequence of a human genome is just the beginning, which is leading to the very complex, very complicated, very, time, money, and labor-consuming duty, which is annotate the genome to find out what is the consequences of each gene, what is function of each gene, and what is the consequences of if we mutated or design or make mutation at one point. So from 2003 to 2008, the 1,000 human genome project was proceeded around the world. Among those, in Vietnam, we have 124 Vietnamese students. KHV stands for King Ethnic in Ho Chi Minh City in Vietnam. Okay? Was put into the 1,000 human genome project. And since 2018, we have 10 countries in the world have reached into more than 100,000 of each citizen was sequenced whole genome. That means that was UK, Japan, China, Australian, Saudi Arabian, US, Estonia, France, UAE, and Turkey. So nowadays we have biological company in Vietnam is not very developed, but there's also some company have started service. If you provide them their money, they can sequence. So if you provide them your DNA sample, they will sequence it and provide you all the related information. For example, based on your DNA sequence, they will predict what is the ratio of you having a cancer or type 2 diabetes, or what is your nutrient absorption. For example, are you lactose tolerance? Are you good at absorbing lipid or protein? And they also provide you sufficient appropriate diet based on your genome. Quite nice. And in the following clip, you will hear a group of science from Italy to develop their project on predicting phenotypic trait and even facial expression from a human genome. So this video from TAP.
In the next 16 minutes, I'm going to take you to a journey that is probably the biggest dream of humanity. Understand the code of life. So for me, everything started many, many years ago when I met the first 3D printer. The concept was fascinating. A 3D printer needs 3 elements: a bit of information, some raw material, some energy, and it can produce any object that was not there before. I was doing physics, I was coming back home, and I realized that I actually always knew a 3D printer, and everyone does. It was my mom. So my mom takes 3 elements. A bit of information is between my father and my mom, in this case, raw elements and energy in the same media, that is food, and after several months produces me. And I was not existing before. So apart from the shock of my mom discovering that she was a 3D printer, I immediately got mesmerized by that piece, the first one, the information. What amount of information takes to build and assemble a human? Is it much? Is it little? How many thumb drives you can fill? Well, I was studying physics at the beginning, and I took this approximation of a human as a gigantic Lego piece, okay? So you can imagine that the building blocks are little atoms, and there is an hydrogen here, a carbon here, a nitrogen here. So in the first approximation, if I can list the number of atoms that compose a human being, I can build it. Now, you can run some numbers, and that happens to be quite an astonishing number. So the number of atoms, the file that I will save in my thumb drive to assemble a little baby, it will actually fill an entire Titanic of thumb drives, multiplied 2,000 times. This is the miracle of life. Every time you see from now on a pregnant lady, she's assembling the biggest amount of information that you will ever encounter. Forget big data, forget anything you heard of. This is the biggest amount of information that exists. But nature, fortunately, is much smarter than a young physicist. And in 4 billion years, he managed to pack this information in a small crystal we call DNA. We met it for the first time in 1950, when Rosalind Franklin, an amazing scientist, a woman, took a picture of it. But it took us more than 40 years to finally poke inside a human cell, take out this crystal, unroll it, and read it for the first time. Well, the code comes out to be a fairly simple alphabet, 4 letters, A, T, C, and G. And to build a human, you need 3 billion of them. 3 billion. How many are 3 billion? Well, we don't really make any sense as a number, right? So I was thinking how I could explain myself better about how big and enormous is this code. But there is I'm going to have some help, and the best person that is going to help me to introduce you the code is actually the first man that's sequencing, Dr. Craig Venter. So welcome on stage, Dr. Craig Venter. Not the man in his flesh, but for the first time in history, this is the genome of a specific human, printed page by page, letter by letter. 262,000 pages of information, 450 kilograms, shipped from United States to Canada, only thanks to Bruno Boding on stage, Lulu.com, a startup that did everything. It was an amazing feat. But this is the visual perception of what is the code of life. And now for the first time I can do something funny. I can actually poke inside it and read. So let me take some interesting book like this one. I have an annotation, it's a fairly big book. So just to let you see what is the code of life. Thousands and thousands and thousands and millions of letters that apparently make any sense. Let's get to a specific part. Let me read it to you. AAG, AAT, ATA. Well, to you sounds mute letters, but this sequence gives the color of the eyes to Craig. I'll show you another part of the book. It's actually a little more complicated. Chromosome 14, book 132. As you might expect. ATTCTTGATT. This human is lucky, because if he will miss just 2 letters in this position, 2 letters over 3 billion, he will be condemned to a terrible disease, cystic fibrosis. We have no cure for it, we don't know how to solve it, and it's just 2 letters of difference for what we are. A wonderful book, a mighty book. A mighty book that helped me to understand and show you also something quite remarkable. Every one of you, what makes me, me, and you, you, is just about 5 million of this, half a book. For the rest, we are all absolutely identical. 500 pages is the miracle of life that you are. The rest, we all share it. So think about again when we think we are different, this is the amount that we share. So now that I have your attention, the next question is, how do I read it? How do I make sense out of it? Well, for how good you can be assembling Swedish furnitures, this instruction manual is nothing you can crack in your life. And so, in 2014, 2 famous TEDsters, Peter Diamandis and Craig Venter himself, decided to assemble a new company. Human Longevity was born with one mission: trying everything we can try and learning everything we can learn from these books, with one target: making real the dream of personalized medicine, understanding what things should be done to have a better health, and what are the secrets in this book. An amazing team, 40 data scientists, and many, many more people, a pleasure to work with. The concept is actually very simple. We're going to use a technology called machine learning. Okay? So on one side, we have genomes, thousands of them. On the other side, we collected the biggest database of human beings, phenotypes, 3D scan, NMR, everything you can think of. Inside there, on these 2 opposite sides, there is the secret of the translation. And in the middle, we build a machine. We build a machine and we train a machine, well, not exactly one machine, many, many machines, to try to understand and translate the genome in a phenotype. What are those letters and what do they do? It's an approach that can be used for everything, but using it in genomics is particularly complicated. And little by little we grew and we wanted to build different challenges. We start from the beginning, from common traits. Common traits are comfortable because they're common. Everyone has them. So we started to ask our question: can we predict height? Can we read the books and predict your height? Well, we actually can, with 5 centimeters of precision. BMI is fairly connected to your lifestyle, but we still can. We get in the ballpark, 8 kilograms of precision. Can we predict the eye color? Yeah, we can. 80% of accuracy. Can we predict the skin color? Yeah, we can. 80% of accuracy. Can we predict age? We can. Because apparently the code changes during your life. It gets shorter, you lose pieces, it gets insertions, we read the signals, and we make a model. Now an interesting challenge. Can we predict a human face? This is a little complicated, because a human face is scattered around millions of these letters, and a human face is not a very well-defined object. So we had to build the entire theory of it. We had to learn and teach a machine what is a face, an embedding, compress it, and if you are comfortable with machine learning, you understand what is the challenge here. Now, after 15 years after we read the first sequence, this October, we started to see some signal. And it was a very emotional moment. So what you see here is a subject coming in our lab. This is a face for us. So we take the real face of a subject, we reduce the complexity, because not everything is in your face. Lots of features and defects and asymmetries are coming from your life. We symmetrize the face, and we run our algorithm, okay? The results that I show you right now, this is the prediction we have from the blood. Now, wait a second. In these seconds, your eyes are watching left and right, and your brain wants those pictures to be identical. So I ask you to do another exercise, to be honest. Please search for the differences, which are many. The biggest amount of signal comes from gender, then there is age, BMI, the ethnicity component of a human, and scaling up over that signal is much more complicated. But what you see here, even in the differences, let you understand that we are in the right ballpark, okay? That we are getting closer and is already giving you some emotions. This is another subject that comes in place, and this is a prediction. A little smaller face, we don't get the complete cranial structure, but it still is in the ballpark. This is a subject that comes in our lab, and this is the prediction. So these people have never been seen in the training of the machine. These are the called held out set, okay? But these are as well people that you will probably never believe. We are publishing everything in a scientific publication, you can read it. But since we are on stage, Chris challenged me, I probably expose myself and try to predict someone that you might recognize, okay? So in this vial of blood, and believe me, you have no idea what we had to do to have a blood now here. So in this vial of blood, there is the amount of biological information that we need to do a full genome sequence. We just need this amount. We run this sequence, and I'm going to do it with you, and we start to layer up all the understanding we have. So in the vial of blood, we predict that is a male, and the subject is a male. We predict that is 1.76 m, the subject is 1.77 m. We predict that is 76, the subject is 82. We predict his age, 38, the subject is 35. Predict his eye color, too dark. We predict his skin color, we are almost there. That's his face. Now, the reveal moment is that the subject is this person. And I did it intentionally. I am a very particular and peculiar ethnicity, Southern European, Italians. They never fit in models. And it's particularly that ethnicity is a complex corner case for our model. But there is another point. So one of the things that we use a lot to recognize people will never be written in the genome, is our free will. Here's how I look. Not my haircut in this case, but my beard cut. So I'm going to show you, I'm going to just, in this case, transfer it, and this is nothing more than Photoshop, no modeling, the beard on the subject, and immediately we get much, much better in the filling. So why do we do this? We certainly don't do it for predicting the height or taking a beautiful picture out of your blood. We do it because the same technology and the same approach, the machine learning of this code, is helping us to understand how we work, how your body works, how your body ages, how disease generates in your body, how your cancer grows and develop, how drugs work, and if they work on your body. This is a huge challenge. It's a challenge that we share with thousands of other researchers around the world. It's called personalized medicine. It's the ability to move from a statistical approach where you're a dot in a Gaussian to a personalized approach, where we read all these books and we get an understanding of exactly how you are. But it's a particularly complicated challenge. Because of all these books, as today, we just know probably 2%. 4 books of more than 175. And this is not the topic of my talk, because we will learn more. There are the best minds in the world on this topic. The prediction will get better, the model will get more precise, and the more we will learn, every time we will be confronted with decisions that we never had to face before, about life, about death, about parenting. So this conversation, and we are touching the very inner detail on how life works. And it's a revolution that cannot be confined in the domain of science or technology. This must be a global conversation. We must start to think the future we're building as a humanity. We need to interact with creatives, with artists, with philosophers, with politicians. Everyone is involved because it's the future of our species. Without fear, but with the understanding that the decisions that we will take in the next year will change the course of history. forever. Thank you.
Okay, so I think it's the end of today's section. So a quick recap in Vietnamese. Ok, chắc là hôm nay thì cái slide này là cái slide quan trọng nhất của ngày hôm nay mà nhiều bạn quan tâm, đúng không? Một chút recap bằng tiếng Việt để bạn nào mà vẫn còn đang ngơ ngác vì Tết thì có thể hiểu hơn. Ok, như vậy là cái điểm của bạn sẽ được đánh giá bằng 20% điểm